So our next speaker is Helena Crowell. She did her PhD in Zurich, at the University of Zurich, within the group with Mark Robinson on benchmarking single-cell methods and spatial methods. Then she, luckily for us here in Barcelona, she moved to Barcelona at the National Center for Genomic Analysis in the group of Holger Hein, where she's been doing all spatial omics stuff. And she's going to tell us about her last work. So thanks for accepting the invitation, Helena. The floor is yours.
Right. Thank you. Okay, so in the title of my talk, there's sort of two key terms highlighted in bold here: colorectal cancer that we want to study and whole transcriptome imaging. And just to make sure we're all on the same page, I want to just start out with how are we able to image the whole transcriptome and how do we get there? So a couple decades ago, this is not working, people came up with single molecule fluorescent in situ hybridization, which is pretty old already, where we iteratively image mRNA molecules or other types of molecules by targeting them with something that's fluorescently active. We take an image, we wash off, and we do another round. Now, the whole transcriptome is maybe a couple 10,000 targets, so this is not good enough. It scales linearly, and we can maybe resolve tens of molecules in this way. So about a decade later, then, people had this brilliant idea of using barcodes. So for example, we pre-assign RNA B to be red and green. In the first round, we target it with something red, second round green. We can stack those images, decode the series of colors we observe in one location, map that back to our catalog, and then see which RNAs were in which location. So now we've gone to exponential scaling, and we can resolve hundreds of targets in this way. Still not good enough. But theoretically, this can scale, to anything we like, but we run into a lot of problems. These barcodes, if we make one mistake, we end up with a wrong RNA, and our image just becomes overloaded with fluorescent signal. So the authors of Merfish in 2016 expanded this idea, we call this error-robust barcoded fish, to use sparse barcodes, meaning that we don't target every RNA every single round. We have some rounds of being black. And secondly, they're distant, so that even if we miscall one of the colors or we miss a spot, we don't make a mistake still. So there's some distance between these barcodes. And this idea is in fact what underlies today's commercial technologies of imaging-based spatial transcriptomics. CosMx, MERScope, and Xenium all rely on this principal idea. And this is a picture of my colleague and I here in Barcelona in front of the first CosMx instrument installed worldwide in Barcelona in Spain. And where I work, we have a lot of technologies for single cell and spatial. It's a research center, but also a service institute. And the projects that I've been involved in the spatial realm, these are just some examples. So one project was more on a biotechnology side to show that we could image monolayers of cells and quantify their RNA and protein instead of sequencing, which scales to millions and billions of cells at the fraction of the cost of sequencing. I've also worked on data, CosMx data on tonsil, colorectal cancer, and now we're acquiring data on a tissue microarray of lymphoma from Barcelona cancer patients. So here, I'll focus on this project, and at this point, I would like to acknowledge all of the people that were involved here. So the data comes from a hospital in Belgium in the lab of Sabine Teschba, who's a brilliant clinician also in actually seeing patients. These samples were shipped to Seattle as part of a pilot study when the technology was still in the research and development part. And they also shipped samples to us to acquire single-cell data. And special thanks to Irene, who is the pathologist in our lab, and a lot of the images you will be seeing are her work, and Anna Pascual-Regant, our spatial group leader, who has been really instrumental throughout. Also, actually, Brooker, formerly known as NanoString, has been very supportive, and Evelyn helped us a lot with doing things that were not possible before the technology was commercial. All right. So this is one of the sections we'll be looking at. But it's not just any colorectal cancer section. This was selected from a huge course of thousands of slides to contain, on a one-dimensional plane, 3 different domains. A healthy reference kind of in green, this is healthy epithelium. Then these sort of transformed pre-malignant lesions and cancer. And those were the sections we were interested in, not simply cancer biopsies, right? And if we then zoom in to one of these areas here, even if you're not a pathologist, which I'm certainly not, you can see the difference between this. These are healthy epithelia, there's a lot of immune cells in between, and then here they become very thick, not monolayered anymore. The immune compartment is completely removed, things become denser, and they just look different, right? So this transition is what we're interested in, and how eventually we go from this to cancer. And chances are that in this room, a lot of people have this state, but it typically doesn't. But it may evolve to become cancer, it may evolve to be treatable, it may be very quickly killing you. And lastly, I want to look into what happens next. So cancer cells can travel through blood vessels or the lymphatic system to other organs where they have the potential to metastasize. And this will, for this talk, be sort of my table of contents. So I want to look at what happens here, what happens at invasion events, and what happens at the metastases. All right. So this is just some numbers. I'm not going to read those out loud, but just to say we acquire fluorescent stains, we have whole transcriptome, we imaged about 4 square centimeters of tissue, and we also acquire single cell data on adjacent sections. And this is then what that looks like. So these are the protein markers, nuclei, epithelia, macrophages, immune cells. These white dots here is one individual mRNA species. And then we can assign those molecules to cells and then sort of visualize gene expression and cell type annotations the way we would do in single cell. But instead of a t-SNE or UMAP, this is the actual tissue, right? Okay. So that's, for the pilot run, this was our initial cohort. We have these 9 sections. And the first thing our pathologists did was to annotate them into these 3 categories that I explained earlier: healthy, pre-malignant, and cancer, across these sections. And she then went ahead and classified the different colorectal cancers more in depth by grading them, so that's the differentiation state of the tissue, staging them, that's how deep does the cancer invade into the tissue. And she also annotated a bunch of other features such as inflammation, angiogenesis, and the growth pattern of the cancer. And all of this was then kind of put together in this table of the 27 regions, where for the cancer regions we have a lot more information. And we can then put them into this 2-dimensional space of grading and staging, where if you're here, you're more fatal than if you're down here, right? Okay. So this was the pathology groundwork. On the transcriptomic side, the first thing that I basically did was to extract profiles from the single cell data that we acquired and use that to cluster the data at a very broad level. We then automatically pool these into 3 major compartments. So for example, B cells, plasma cells, T cells go to immune, endothelia, fibroblasts go to the stromal compartment. And we then split these and recluster, which gives us a much higher resolution. So now in the epithelia compartment, for example, we see very nice patterning here. We start seeing these gradients, and here these 2 fetal populations that form the outer ring of this cancer lesion, and the stem-like population here, that's kind of the tumor core. All right, so if we take all the histopathology and just these clusters, there's a lot of things we can do already. And one thing I really like is kind of taking one table and visualizing it in dozens of different ways and seeing what we get out of that. So here's a very simple plot. We just compare the composition in terms of the compartments across the regions. And we see if we're healthy, we have a lot of immune cells. And then we go towards having more epithelia, less immune cells, and in the cancer we tend to also get more stromal cells. If we just flip the aesthetics here, we can see that these are the subpopulations colored by basically their composition in terms of the regions. And some populations like this one are really exclusive to cancer. This one is exclusive to TVA, and these are found largely in the healthy tissue, right? So this helps us a lot to understand and annotate all of these different cell types. Now, we can take just one section and compare again the same things, but across the different compartments. And we see here, this is the epithelium. The regions are totally different. There's drastic shifts in the epithelium compartment. Immune and stroma are more subtle. We kind of lose some plasma cells. We gain tumor-associated macrophages. The stroma compartment has some shifts, but it's much more subtle. Now, let's take all of these cancers and compare them across the different patients. And if we do that, we kind of see here these roughly two groups. It's not fully accurate, but basically these are ordered by the grading from the pathologist, and we end up with these two groups of a fetal-like endpoint and a stem-like endpoint. And for those who know a bit about cancer, we expect cancer cells to de-differentiate. They don't carry out the function they should. That's why these stem and fetal-like states. And eventually they will simply proliferate and do nothing else, right? And these two endpoints seem to be different in their prognosis and the grading and the outlook kind of the patient. And I'll come back to that later. Now, at this point, we've done some comparisons, very discrete, but we have not considered space in any way. And the way we do that a lot of times is we take a cell and we look at its local microenvironment, 50 microns, which contains maybe 10 to 30 cells. We can then count things, how many epithelial cells are there, how many immune cells are there, or we can quantify the relative composition of the microenvironment. And this gives us then sort of local estimates of cellular density and entropy, which we can again compare in a dozen different ways, right? So these are then these density estimates we get across the tissue. Every single cell, we take the neighborhood, we count, and we can plot that in space. And we see, for example, here in the immune compartment, this box here of high immune density, which marks a tertiary lymphoid structure. In the stromal compartment, highest density, kind of where the basal membrane is of the tissue. Okay. Now, if we take these estimates now and compare them across the regions, we see in the epithelial compartment this increase in density towards cancer and a decrease in density for the immune and stromal cells. And in terms of entropy, this is now across all sections. We see that the entropy seems to be lower in cancer, kind of intermediate in the TVA state, and highest in the reference. And what this is telling us is that cancer cells tend to be surrounded by themselves, by cells that are just kind of the same, and also doing the same. They tend to just be denser and more homogeneous, and there's not a lot of heterogeneity, and immune cells and stromal cells get entirely removed in favor of dense epithelium, okay? Now, these are a bunch of box plots. I can tell you that each of these contains about 100,000 cells, so this is a very rough picture of what's happening, and we want to arrive at a more continuous view of this transformation from healthy to pre-malignant to the cancer that's like happening here, right? And how we got there is a bit of a long story, but let me give you maybe a toy example. So this is the decathlon data set in R. Every point is an athlete, and they're colored by the time they took to run 100 meters. And this is the first two principal components, okay? If we take now these numbers and we rescale them from 0 to 255, we can plot that in color as an RGB representation. And what we then get roughly is a red cluster, a blue cluster, and a green cluster. And these three principal components separate essentially people that are good at running, that are good at jumping, and that are good at throwing things, because those are the three types of disciplines that exist in a decathlon. In our tissue, the principal components represent the three major compartments. The first one is the epithelium, the second is the immune compartment, the third is the stroma, and sometimes they kind of mix together and we see different shades of red, green, and blue. We can then repeat this and just pull out the epithelia, and that's where we start seeing these gradients here towards the outer rim, and this darkest region here where the cancer is, right? So that's interesting. It means the principal components capture the transcriptional variability as we go from healthy to cancer. And there's a method that fits through this space in higher dimensions, a curve, and projects cells onto this curve to estimate the distance along this sort of gradient, and that's slingshot. And this is what we ran here. So if we do a slingshot and we get this pseudo-time, or I think of it as transcriptional development along this line, this curve, we get this very nice gradient, and we can again take every cell and ask what is around the cell. We move to the next time point, what is around the cell, and we kind of inspect the local microenvironment along this trajectory. And this lets us then do a lot of cool things. I'm not going to go into the biology too much, but we can quantify these dynamics of how the epithelial compartment changes and the immune and the stromal compartment. And we can also quantify locally functional pathways like glycolysis or inflammation and intercellular signaling. And in terms of results, this is really not a lot different than these heat maps and box plot, but it's a much more continuous view instead of kind of throwing all your cells together in large buckets. Okay. So at this point, I think about 1 year into the project, our pathologist, one day she came and she said she found something cool. And she pulled up her screen and she showed us something like this in a pari where she had this crypt, that's one colonic crypt, where half of it is TVA, and one cell later there's cancer, right? So it's completely split. And she didn't know, or nobody really knew, if this is because the crypts kind of fuse together, or there's an abrupt shift, like the cell just jumps to another state, and then you kind of have cancer. And as she then spent about a week on this, she found 42 of these events, not just between TVA and cancer, but also from healthy tissue to cancer. Like one cell is healthy and the next cell is cancer, and suddenly the rest of the crypt is cancer. And we then looked at this in the spatial transcriptomics data. I will zoom into this in a second, but essentially we can see the selections that we have. We expand them by a 50 micron polygonal expansion, and we then can look at what is the immune epistromal annotation, what is the cluster annotation, and what are the pseudotime estimates in these scripts that she annotated. And these are just 4 examples of the 4 different cases. And what was, for me, I thought it was really astonishing, is that even though we cluster 3.5 million cells, we get these scripts here down to very few cells. So the transcriptional clustering is very accurate in recapitulating these local events of jumping from one state to another. Okay. Now, to finish kind of this transformation bit, if you remember, the next step that can happen in cancer is that we have these cancer cells, and we have nearby blood vessels and lymphatic vessels, and if the stromal compartment gets transformed and interacts in a certain way, these cells can enter into these vessels and kind of cross that barrier, travel to other organs, and given the right microenvironment, they might metastasize. So we also found these type of events in the section. This is an example of an invasion where we have a vessel, and inside there's epithelial cells, and down here we have a vessel where there's no epithelial cells inside. And what you can immediately kind of see is that it's kind of the same cell type inside, the purple one, and there's these blue guys that are forming the vessel, right? And again, we can go ahead and identify a lot of these events across all of the different sections. And this lets us then quantify what is happening at these sites. And these are all of the blood vessels. We tried to make sense of it, but it seemed pretty random. Meanwhile, the invasions are very consistent. So we always have this purple cancer population that is invading the vessels. We always have a lot of tumor-associated macrophages and neutrophils. And we always have these blue cancer-associated fibroblasts that are around it. And why that's interesting is because we can now go ahead and we're doing now validation experiment on the protein level to mark these invasion events, because this would really tell us what type of microenvironment allows cancer cells to invade and to metastasize. Okay, now, metastasis, we had one. This is a lymph node section that we had. So if you have invaded, you can metastasize. And this section here had 2 metastases that the pathologist annotated. And surprisingly, even though this is the same patient, they're completely different. So the growth patterns of these 2 metastases on a histopathological level are completely different. And the primary tumor of this patient also has completely different cancers. So this red tumor here mirrors the growth pattern of the first metastases, and this one here mirrors the growth pattern of the second metastases. So we have one primary tumor with sort of 2 different cancer regions and 2 different metastases, right? So on the transcriptional level, we wanted to kind of, dig into that. So we first gated for CD45 negative pan-CK positive cells to select for epithelia, and these should not be present in a healthy lymph node. And secondly, we take the cluster annotations of the primary tumor to extract reference profiles, right? Because we think these cells come from this. And if we then annotate the metastatic cells, we again find completely different transcriptional profiles. On the one hand, again, this fetal state, which we found to be more deadly. And on the right, the stem-like state, which tends to proliferate, but it's less malignant. And why that's, I think, interesting, and particularly Sabine at the hospital was interesting, is it means you can have a patient with metastases where one will kill them and the other is treatable, or you might need different treatments for these. And that's certainly something that's interesting to understand and to be able to detect. Okay, so just to summarize, we have acquired a lot of data. We do an in-depth histopathological characterization and clustering, and these 2 things combined lets us already do a lot of things. And there's another part of the analysis that I sort of skipped over. We can get a more continuous view of this transformation through an epithelial trajectory that lets us quantify local dynamics, and we trace this down to individual crypts of maybe a few hundred cells. And lastly, we trace then invasion events to metastases with divergent fates, both on a histopathological and also a transcriptional level. And if I look back at this, project, I always thought it's very beautiful data. And one thing it's taught me is that if you have this type of data on your screen, you can really do a lot. Like you can do any method you want, and there's thousands of methods now out in the spatial field that you can try out, and they all seem exciting. But often the questions that the hospital asks and the biologist asks are very simple. They will ask you, what's the distance of this? Or can you plot this versus that? Like, this is what they want to know, and this is what they're interested in. So you kind of need to provide things that are simple and understandable for them to understand, right? So I think my take-home message and my lesson learned was that if you know the basics, you can really do a lot, because I would argue that all of these analyses were mostly done with data wrangling, summarizing, and plotting things in different ways and subsetting certain things. And that, I think, was for me more important than being able to run some fancy kind of computational methods on the data. Okay. And just to say that all of this is publicly available. We're sharing the H&Es, the immunofluorescent, the whole transcriptome data, all of the results as R, Python, and language agnostic downloadable kind of files. The histopathology was done in QPath, immunofluorescent in Apari, and all of the analysis was done in R. And all of this is put together into a SnakeMake pipeline that kind of lets me process all of these sections in parallel and do lots of things on the cluster. I hope also that maybe there's no package, but I hope that people can maybe draw some inspiration of the types of analyses we did in the code. And with that, I just want to mention that one thing I completely skipped over is kind of all of the pain that was involved here. So this data, when it came, was a pilot study. Nobody had analyzed this. And I really felt like this is not the same as single-cell RNA sequencing. And you see that in every analysis you do, it just doesn't work. So first we start by imaging. We have 64 kind of things, 4 times 16, a stack of images. We then call each of these molecules, all of these images get overlaid, and then we decode kind of what RNA comes from there. And finally, we segment and assign them to get the count matrix. And this is just very different than what you do in next-generation sequencing to get your count matrix, right? And all of these things have a lot of problems. So the CosMx runs for about 2 weeks. Things don't bind where they should. Here, you would just make a lot of mistakes. You miss something, you miscall something, right? This is, I think, the biggest problem, is that this assignment here is not accurate. It's a 2-dimensional image of a 3-dimensional system. You're misassigning transcripts all the time. And then here, if you try to plot the mean variance of this, it's crap. There is no mean variance relationship. You don't know how to model these counts. You don't know how to do quality control. And I think, the main takeaway for me is like you cannot just run normal methods on this data. And a lot of work, I think, is required to kind of improve on this. And lastly, on the other side, I think there has been a lot of development to the right of the schematic in terms of methods. And in Bioconductor, throughout this year, there's been a great multi-institutional, cross-continental effort. I think we span 3 continents, and the neutral Switzerland, where we sort of, mostly online, people worked on a lot of tasks that they had expertise in to put together this online resource, OSTA. You can find the link here. Also, shout out to Estella for these beautiful stickers. And there will be a workshop tomorrow by Alice Patrick and Estella. So if you want to work a bit on spatial transcriptomics data, I recommend it. And with that, I hope I have time for questions, and I thank you for your attention.
Thank you, Helena, for a nice talk. So the way I see it, there are kind of 3 big elements in all this data analysis beyond the interpretation. There is annotation by the histopathologists, there is the data generation, you mentioned 2 months, and then there is the data analysis. So how much time do these 3 aspects, these 3 parts take? Which one is the most time-consuming? And what's the potential for automation for these 3 parts?
Yeah, that's a good question. So the planning, I think we start maybe 6 months before we do a run to discuss it. And then it's about the hospital has to select things, they have to send it somewhere, they have to select these samples, ship them somewhere, then you need to order the reagents. So it's like 6 months from discussing it to doing the experiment is a regular timeline. Then I think majority of this analysis I did in 2 weeks. 2 years is looking at plots and then changing the quality control, and then doing it again, and then looking at it, and I think that's 2 years, is really the annotation. But then even when you have an annotation, the biologists, they like sit down and they have to look at all of these signaling pathways. What could that mean? Then they do literature search. They read papers of what's been shown 50 years ago and it's already known, or what is maybe a recent paper that could support this. So I think that's what's the bulk of the work, is the interpretation. Yeah.
Thanks a lot. It's the most beautiful spatial talk that I have ever seen. I have a question about your cosmics technology. I have seen already several times that the imaging is happening like in segments which you stitch. And when you show your pictures, you see clear the small segments, meaning that I don't know how you process it because the signal is somehow not homogeneous. How do you deal with this?
So I think the xenium stitches, the cosmics does not. You get like even white space sometimes between the fields of view. We do just some quality control at the borders because we know the cells are just worse and they might be duplicates of the next kind of field of view. So we do some quality control. And then I think the main thing is that in most of the computation, the problem arises if you want to quantify distances or neighbors and you're crossing over into another field of view. So I think by kind of looking at radial neighborhoods, you think that by doing this over 3.5 million cells, your results are not going to suffer from the few cells that are kind of at the borders. You know, it kind of crosses out. But right now, there's no computational solution to this. It's just noise that is in your results, and you hope that it's not going to affect what you find, yeah. But the cosmics doesn't stitch. It's really annoying. It's horrible, actually, yeah.
Okay, excellent. I'm afraid we're on time for a coffee break, so you can catch up with Helena at the coffee break. Let's thanks again, Helena, for her talk.